I'm glad to be here to share some of the work that we've been doing at MIT related to AI for chemistry. So we think a lot about this intersection because we sit at this interface. My group sits at this interface between chemical engineering and electrical engineering and computer science as part of MIT's Schwarzman College of Computing. So we're going to talk about a couple of threads and themes that we work on in the group, and in particular, try to highlight, right, not just the technology, but also what we're trying to do with this technology and some sort of small comments, at least side comments on the potential for dual use, because I think these topics are, of course, of interest to many folks in the room. Now, obviously, we're at a moment where we're seeing an increasing use of AI across the chemical sciences, across science in general. And we see a lot of high profile examples like AlphaFold, of course, receiving a Nobel Prize in chemistry, right? Common day, sort of usage of tools like large language models. But then you also have AI being used across the sciences in more narrow areas, right? Areas that you wouldn't normally think of, right? Weather forecasting, trying to solve very large PDE systems and simulate climate and weather events. People use it to control reactors, control instruments. Of course, there's a lot of applications in biology and biomedicine. We think about its intersection with chemistry, as you'll see. But what unifies the applications across the sciences and how we think about AI broadly is its role in the scientific process. Okay? So we typically will think about using AI to help us make sense of the world, analyze data and interpret the results of experiments, and hopefully come up with new hypotheses and new ideas that will drive future experimentation and future discoveries. So in chemistry, what is it we're trying to do? We're trying to drive scientific discovery. And what that means for us is we're trying to either find new physical matter. So that could be a new molecule, that could be a therapeutic, that could be a new material that enters into some solar cell. We could also be trying to discover a process. So often the result of some discovery campaign could be a manufacturing process or a synthetic route is the discovery of models, right? There's very empirical models like trained neural networks, which count as perhaps discovering a model that describes the relationship between structure and function. But of course, models can be more abstract and higher level. And while I'm not going to talk about it, and there are very few examples in the literature, it's interesting to think about the way that AI could help us invent these more abstract, fundamental notions of models, things like the periodic table, right? Or sort of orbitals, discovering those from data. But we're going to be a little bit more sort of down to earth in this talk, thinking about the discovery of molecules and synthetic processes to make them. So small molecules is this really nice modality. They make up the majority of our FDA-approved medicines. We also use them in agriculture. We use them in materials. We use them in defense, right? They have this really interesting broad set of applications just by rearranging, the same familiar sets of elements, right? Carbon, hydrogen, nitrogen, oxygen gets you most of the way to covering a lot of these applications on the slide. And what's interesting is that, just by rearranging these same kinds of atoms, you can access such a broad range of function. And this is really because chemical space is huge, right? If you think about all the possible ways you could arrange these atoms in, there's an enormous number of possible chemical structures. And people estimate this number, various ways. So I've, 10 to the 30, 10 to the 60, it becomes meaningless, right? Because it's virtually infinite, right? We can't exhaustively search this space. And so, of course, we have to think about ways to accelerate the process. And so I see this sort of enormity as both an opportunity, because in principle, there exist chemicals within this space that achieve such a broad range of functions that are useful for medicines and more. But of course, it's a hard search problem. So it's a search problem that we tend to approach then with this sort of classic iterative workflow, right? We don't think that we can anticipate, exactly what molecule to make without some experimental testing, some iterative feedback. And so we end up finding ourselves in this empirical loop where we might design some molecules that try to satisfy some goal we've set out. Maybe that goal is some set of properties we want to achieve. After designing those structures, we'll need to worry about how to synthesize them, right? Unlike many applications of AI where you design maybe images or text, the things that we design needs to exist in the physical world. And so we have to physically instantiate the molecules we come up with in order to test their properties experimentally. So that comes into play in this validation step, and that validation data feeds back into our models. Those models then help us presumably design what comes next. And so we like to think about lots of different opportunities to accelerate this process and to bring computation into this process to support all the complicated decisions we have to make, right? We have to decide what we want to make, how we want to make it, right? Justify why we've made it. And then when we get new data, what do we do with that data? And so just walking around the cycle, starting at the bottom, we can think about this sort of piecemeal use of artificial intelligence helping us answer questions like, what are the properties of this molecular structure that I've just hypothesized? We can think about the design question of just directly saying, what molecules should we be trying to make or test? We can think about models that try to come up with the synthesis plans or the recipes for how we might produce those structures. And overall, this will help us accelerate, our navigation through this cycle. That's the idea. And a lot of the projects in my research group at MIT touch on different aspects of this workflow and different opportunities to speed up this process. And this will kind of frame the rest of the talk, right? Walking through this cycle, starting first with brief comments maybe about the modeling side and how we think about using structure property relationships to help us sort of know what molecules are worth making. This goal of relating molecular structure to function is not new, right? We've been trying to do this for decades and decades. It was sort of formalized arguably in the 1960s. And the idea at the time, the idea still, is that we want to correlate aspects of a molecule structure with some property. That property could be a physical property like a solubility, it could be a biological property like potency or binding to a specific protein target. It could be sort of another chemical property like reactivity. There's any number of things that we could try to predict about a molecular structure. Now, the relevant sort of change that's happened in the past decade that's really been accelerated by machine learning and deep learning in particular is a transition from the sort of paradigm where we would choose to represent our molecule through a set of sort of expert defined features or a fixed numerical vector describing aspects of that structure to a paradigm now where we're trying to learn the representation directly from the original molecular structure, perhaps here represented as a graph. So we've always tried to be very clever, right, as a field in doing the second learning step. So how do we take a vector description of a molecule and predicts a physical property? We've applied machine learning to this for decades. The change since about 2015 is that now we use things like graph neural networks to take a graph structure, learn a numerical embedding, and progress that sort of ends to end with properties of interest. And so out of MIT, we developed, in collaboration with many others, this toolkit called ChemProp. So it's just sort of continuing the development of graph neural networks. It's just a particularly, I think, convenient off-the-shelf solution with a good set of hyperparameters to go through this sort of message passing operation that sort of is emblematic of graph neural networks. And it's just a convenient way to say, I've got a data set of chemical structures and properties, and I would like to learn that relationship. So I'm not going to say too much about this class of models because property prediction models, again, they're ubiquitous, right? You can't avoid them when you think about molecular design. I will comment, right, that there's always room for improvements, and we think about some of these where we'd obviously love our models to use less data. We'd like to be able to say, okay, here's, 3 structures and maybe their biological activities. How do I generalize to, 10 to the 60 candidates? We'd love to be able to generalize from less data. We try to build techniques that quantify their own uncertainty. So if we have models that don't see that many unique chemicals, they can at least tell us when they're confident or unconfident in the predictions that we ask them to make. We are still in this state where these are all sort of imperfect, right? We can't generalize perfectly to new structures we haven't seen before. And so importantly, right, the sort of bottom left sort of sentence pointing out the fact that we're always almost going to rely on some degree of experimental testing and feedback, right? You can't really escape that iterative design cycle. At the end of the day, we're going to need to test compounds in the lab and use that information to iterate. And so that sort of brings us already to the sort of next step in the process, thinking about, well, okay, if we have some models that can predict some structure property relationship, how do we approach then the design of structures or the selection of structures to take into the lab and to test in this loop? So we have this hypothetical relationship that we maybe approximate with machine learning models that tells us how structure relates to function. We can draw it in this cartoon surface where as you change the structure, the X and Y axes, you change the property from good to bad. And the idea of design is that we'd love to find the molecules that live at the maximum of this function, right? We'd really like to find the structures that will achieve that maximal binding potency, right? Or maybe that maximum, quantum yield if we're doing some photoelectronic application. So we can do it with our structure property model is we can screen vast chemical spaces. So we could say, here's a list of candidate molecules. I can go online right now and I can download a list of 2.7 trillion molecules that Enamine is willing to sell me. And I could say, which of these 2.7 trillion molecules has the best predictive property? That could be the basis for how I drive then my set of experiments. And so virtual screening could just say, for this candidate, here's the prediction. For this candidate, here's the prediction, and so on. And that can work, and that's been the dominant paradigm. But there's been a lot of interest, of course, in using generative techniques instead. I'm thinking about the role of generative AI and generative models to propose novel structures, novel chemical structures that don't belong to that fixed virtual library. Because the promise or the hope is that if we have some understanding of a structure property relationship, we can try to invert that relationship and go from the desired property directly to the structures we want. So we could hopefully, right, the goal would be to say, I have a generative model and it's going to propose, the chemical structures directly that live at these maxima, or maybe it takes this sort of nice smooth path up the hill to find these optimal molecules. So I'm going to talk a little bit first about the virtual screening sense, because this has really sort of changed the way that people approach molecular design in pharmaceutical applications, because we have reached a point where chemical vendors list out billions and trillions of molecules that they're willing to sell you, right? This is different than how things were, 5, 10 years ago. So these chemical vendors and companies as well to build what we call make-on-demand libraries. So these make-on-demand libraries basically say, I've got some finite number of molecules in my laboratory, right, that I have access to. And I have some set of robust chemical transformations that I think I can run. So I think I can take an acid and an amine, make an amide bond, make that amide product. And so you define this sort of small set of transformation rules that you believe that you can run, and you enumerate all the products that you think you can make in 1 or 2 or 3 synthetic steps. And this maps out billions or trillions or more of hypothetical structures that they believe they can access, that they're willing to sell you. Now, navigating these large virtual spaces is not trivial, right? Because you have billions or trillions of candidates. And so we think about ways to do discrete optimization or Bayesian optimization in this discrete space to accelerate the search process. So with these large libraries, one of the ways that we screen them with application to drug discovery is using basic structure-based drug design methods. So really coarse tools like docking, they try to approximate the binding affinity between that small molecule and a protein of interest. They're really not very good, but they're an okay sort of first filter that can be applied at large scale. And so typically we would just take our large set of candidates and we would exhaustively screen that candidate. And then we would say, for these 100 million molecules, what are the 100 million predicted binding affinities? And then you'll sort your list to find the best candidates and maybe take those into the laboratory. But you can just insert a simple model into this workflow. And you can say, I don't want to do 100 million simulations. I'll do a few, maybe tens of thousands. I'll train a surrogate machine learning model. That machine learning model will not be predicting the results of an experiment, but it'll be predicting the results of a simulation. And so we skip the simulation. We use the model to tell us where in chemical space should we look and where in chemical space is it not worth looking. And so we can build a lot of different tools on top of this. So we can use our imperfect uncertainty quantification to, again, eliminate regions of chemical space and say, it's not worth searching these billion compounds over here because they all look bad, right? So we use models to steer, right, in this gigantic search space of molecular structures. You can extend this quite easily to multi-objective optimization. So we can do proto-optimizations if you have multiple properties of interest, as we often do in molecular design. And recently we've been working on different sort of acquisition functions in this Bayesian optimization context that try to sort of mitigate this risk that you do this virtual screen or you do this evaluation, you pick molecules to test and they all fail, right? That can happen, right? You use your imperfect simulations to select molecules to test and they might all fail. And so we think about building sort of acquisition functions that diversify that selection on the basis of model uncertainty. And so happy to talk more about that later if folks are interested. But this is sort of just virtual screening. And of course, I mentioned generative design being of high interest, and it's of interest to us as well. Again, the promise is that we can use models to invent new molecules with better and better properties, right? So maybe it's a more potent drug. The way that we typically will use generative models is we will have our generator itself, and we will have some sort of evaluator or feedback mechanism that takes an idea of a molecule and scores it. It'll say, that looks good, that looks bad. It's a property prediction model. So we still rely on that feedback mechanism. It could be an experiment, it could be a machine learning model, and it could be a simulation. But if we have that kind of feedback, we can build a number of different types of generative architectures to invent new structures. And so this animation is just showing one model that thinks in terms of graphs, building up atom by atom, a molecule that's predicted by a separate function to potentially be a protein degrader. Now, is that a good property predictor? Potentially not. But given that property prediction, this generative model is inventing structures that it observes to optimize. And so this is fun. I think it's fun, at least. But the problem is that although we want these models to be creative and invent new structures, likely to have unprecedented properties, sometimes when you ask them to invent new structures, they're a little bit too creative. So the kinds of molecules that you can get from these models sometimes look quite absurd. If you're a chemist, they're absurd. If you're not a chemist, you end up with these motifs that don't look like they are reasonable, right? They're not stable, they're not accessible, they really just don't make any sense. For some reason, this one particular model loves putting boron in aromatic rings. You have this sort of very catastrophic failure mode of generative models, where they propose structures that they earnestly believe to be good structures. And maybe your property predictor, because it is imperfect, says, this looks good to me. But this is not something that you would want to give to a chemist or give to, a colleague to try to synthesize. And so this really is one of the sort of big failure modes of generative models that we've worked a lot on addressing over the past few years. But it raises this really important question of chemical synthesis, because the reason why these are bad suggestions is not inherently because boron is in a funny place. It's because there's no way for me to take this idea into the physical lab and test it, right? I can't access this, right? Even if it thinks it's correct, there's just no way for me to make it. And so that sort of brings up this then question of synthesis, and that's a major focus of my research here at MIT. So synthesis, right, is an obstacle to accessing molecules in the physical world. It can be really hard for some structures, easy for others. Synthesis planning is the process of taking a target and identifying some plausible series of reaction steps that can access it. So given this sort of target molecule in the bottom right, I might want to devise a recipe that tells me, here are the building blocks to purchase, the ingredients, here are the conditions to use for the reactions, and you will build up this complexity bit by bit, right? And this turns out to be a linear pathway to access this final structure. So humans are really good at this, right? Humans learn how to do this in organic chemistry classes, take the, organic chemistry exams. Classically, will have you do this sort of retrosynthetic planning where you start with the target product and you have to reason your way backwards. So you typically start from the end point and do this reverse logic and say, well, how can I make this molecule? How can I simplify it? How can I simplify it further until you reach starting materials that you can purchase? This is the basic notion of retrosynthetic analysis. And so this is something that we can also teach computers to do as well. And people have been since the 1960s. And before we get there, sort of comments on the role of AI for synthesis. I view there as being sort of several different ways that we want to use AI to help chemical synthesis. The foundation typically is data, right? It's traits, but it's trait for a reason, right? We want to catalog and understand just like what has been done before, what reactions have been run, who ran them, what was the outcome. And this layer of data is something that we want to make searchable and that chemical publishers have thankfully made searchable over the years. But it's on top of that, we want to layer on that next stage of reasoning. So we want to think about training virtual lab assistants or tools that gain insights from that data and make recommendations to us. They tell us what kind of things we should try. If we run a reaction and the yield is bad, we ask it, or we get some feedback like, why was this bad? What should I do next? So on top of this sort of data layer, we can have this sort of assistant layer, like a co-pilot for the chemistry lab. Now, eventually, we would love to have models that understand the nuance of chemistry and can, run autonomous laboratories, make their own decisions, troubleshoot their own things, and just give us molecules with zero intervention, right? That's the dream of autonomous chemistry in the future. We're, of course, not quite there, but that's where a lot of folks in the field are trying to build to. But we're going to stay on the lower levels and think about sort of the data and the inferences that we build on top of this data. So like I mentioned, publishers have been tabulating data on chemical reactions for quite some time, which is an example from the Reaxys database. It tells us who ran the reaction, what were the reactants, the starting materials, what were the products, what were the conditions, what were the yields. And so this gives us a lot of information to learn useful patterns of chemical reactivity from. So we have this nice example of saying, well, if we want to make that product, we know that's a set of reactants that we could use to make it. And if we want to run this reaction, we know this is a set of conditions that we might be able to use to run this reaction. And so we can build models on top of this, despite the fact that there's a lot of missing information and confounding variables that we don't have. Okay. So there is a lot of information and nuance about chemistry that matters, a lot of nuance about chemistry that is simply missed by these databases. They didn't try to tabulate it, they didn't try to report it. And so there's some things that we can't do with this information. But we can still train models to do things like retrosynthesis using this information source. Okay, so retrosynthesis is starting with the target you want, working your way backwards. So what this requires is we need some model that can take a product and recommend reactants that could produce it in one synthetic step. You can't make a lot of super interesting things in one step. So we need a way of doing this multiple times. This is a recursive search process. As soon as you start talking about this recursive search process and expansion, you start thinking about combinatorial explosion. And so you need tree search strategies to mitigate that search, because as you go further and further back, you have more and more options. And so we need tree search algorithms like Monte Carlo tree search, A-star search, right? Your classics from computer science. These are what we use to try to map out this landscape of options. Of course, we need to know when to stop. So we need to have a database that says, you found something commercially available, now we can stop. And so there's a lot of research being done in the fields trying to improve our ability to map out these hypothetical chemical reactions and do this search process, or do this multi-step search process. And when we apply these tools to relatively simple compounds, we sometimes get a lot of nice, good recommendations. So we can say, I'd like to make this molecule. We can query one of these recommender models, and we can sometimes get at hundreds or thousands of different ideas for how you might be able to synthesize this structure. So even if the models, right, in their training data have never seen this molecular structure, they look at it, they analyze it, they understand what kinds of disconnections can be made and how it maps back to commercially available starting materials. And so we can recommend, right, for these simpler structures, lots of ideas for how we might be able to synthesize them. If we put in something very complex, perhaps we don't get good recommendations or any recommendations, and there are limitations, of course, to these methods. But what we've been trying to do over the years is, as we develop the algorithms that make these predictions and learn about chemical reactions, we've been trying to, in parallel, develop software that is sort of usable and accessible for synthetic chemists, right? If we build a nice command line tool that's not actually terribly useful for practicing synthetic chemists, because most of them do not want to use command line tools, right? And so we've developed different sorts of interfaces, graphical interfaces for using these kinds of software tools, and we've deployed them and open sourced them and have gotten pretty good adoption from the community. There are a few other open source tools out there now. At the time that I started working on these directions as a student, there's really nothing in the open source pre-competitive space for planning synthetic pathways. And so I think now we're seeing sort of a really nice, both open source and commercial ecosystem around these kinds of capabilities. Now, the reality of synthesis is a little bit more complex. Okay. So if I want to actually make a molecule, it's not enough to have that basic design of a pathway of here are the building blocks, here's the basic conditions. You have to make a lot of really specific decisions when you want to physically run a reaction in the lab. Okay, so it's not just going to be the basic sort of abstract plans that we can solve with our synthesis planning tools, but we have to decide details like the orders of addition, your rates of addition, purification, or isolation status. And at some point, we have to decide what hardware do we use as well. The databases didn't tell us to use a round bottom flask or a vial or a well plate. We have to make some decision about what the physical hardware is that we want to use. And this, of course, will have implications for automated laboratories that we'll talk about a little bit later. But overall, right, we're at this sort of nice state in the field where we do have these tools that can plan synthetic routes to many molecules that are on the simpler side. And they can give you suggestions for how you might produce them quite quickly in this nice open source ecosystem. And so we've seen people use these tools for, the classic use cases we intend. Just here's a molecule that I want to make, come up with a recipe, right? That's what they're designed for. But the tools can also be used for some adjacent tasks. There have been some really interesting applications in literature. So people have applied these kinds of synthesis planning tools to shore up different supply chains. So if you have one manufacturing pathway to a drug, if there's a critical shortage of a starting material, you might be able to quickly propose an alternative synthetic pathway to avoid that shortage. There's some companies thinking about how we can onshore building blocks for our pharmaceutical supply chain. So right now, even if you were to sort of synthesize medicines, here, The government is quite nervous about the fact that the starting materials typically come from overseas. And so there's a lot of interest from the government in having, domestically sourced starting materials. People have also been using some of our other models for synthesis to anticipate transformation products and using this for things like analyzing the safety of additives in vaping fluid and sort of recognizing, yes, there's a ton of really bad stuff that you can make and enumerating the possible structures, cross-referencing that with other data to identify what are those molecules that might be byproducts of these processes. Now, in the dual use worlds, right, sort of, I promise that there'd be some comments on this. So you can imagine that if you have a tool that can tell you how to make any given molecule, you can put in molecules that we don't really want people to make, right? You can imagine people making hazardous chemicals, learning how to make regulated substances, the same, capabilities that let you do this supply chain robustness, where you avoid a specific starting material in shortage, you could also use to avoid a specific starting material that's a controlled substance. And so there's a little bit of concern, right, in how people might use these tools to circumvent the way that we regulate starting materials. Now, it's not all bad, right? So there's some mitigating factors to these concerns, right? So one thing, of course, is that if you are making a new molecule that's quite challenging, or you're making a new synthetic route, there's further development and expertise that's typically required to actually make it work experimentally. So it's not just you push a button, you get a molecule out, right? There's additional chemistry expertise. And part of the mitigation as well is there's sort of interesting discussions around, how much do these tools change the ease by which people can access controlled substances? Because it turns out, right, there's a lot of known synthetic routes to known, illegal molecules. And so perhaps these tools don't fundamentally change that landscape, but it's an important consideration to think about the way that access to these programs might be influencing the way that people approach regulating controlled substances. I want to pivot back to now the design question, but this time still thinking about synthesis in design, because this is one of the major opportunities to improve the way that we use AI for chemistry. I alluded to the failure mode, right? When you design molecules with generative techniques, you can sometimes get suggestions that are quite absurd that you can't take into the lab and make. And so the solutions that we've been pursuing for this is to redefine what it is we are asking the generative models to do. So when we use AI to design molecules, the typical way that we will design them is using the language of spile strings. So we can encode molecules just as strings. Maybe we write them as graphs. We like graphs. Maybe we write them as sort of three-dimensional point clouds. And so we use things like language models to propose strings, or graph generating models to propose graphs, or 3D point cloud models to propose point clouds. But the point is that when you design in the space of structure, right, you design a molecular structure, that does not mean it's going to be easy to access. And so our solution for this is that we're going to ask the models to design, instead of structures, design pathways, right, or synthetic processes. And so we're going to build generative models and other types of enumerative models that think about designing molecular structures via the generation of a synthetic plan. So we force the models to say, here are the building blocks I'm going to use, here are the reaction transformations I'm going to use. We don't just let them move atoms around arbitrarily. We constrain them to think in terms of chemical transformations that we believe we can run in the laboratory. Now, the simplest way to do this would be to take a synthetic process or take a reaction scheme. In this case, it's a coupling from the sulfonyl chloride and its arylamine to make this candidate that a collaborator is working on for tuberculosis. And we could say, if I want to guarantee synthetic accessibility of the molecules I'm designing, why don't I just take a whole bunch of, sulfonyl chlorides and a whole bunch of arylamines and just find out all the combinations between them? These are the sort of basic enumerative approaches that chemical vendors use, but it's saying, I'm not trying to change the synthesis. I'm just trying to change the building blocks. And here are the molecules that I think I can access that look similar to this structure. Now, this is sort of has its own implications for sort of dual use concerns, and just the ease of accessing novel chemical structures, I think, is something that is underappreciated. We did an analysis some time ago, just looking at the sort of basic enumeration capability. How easy is it to generate analogs? It's very easy to generate analogs of structures. So you can read open access scientific publications on synthetic pathways to molecules. In this case, this is fentanyl. It's a very straightforward chemical synthesis in the grand scheme of chemical syntheses, right? And what you could do, right, is you could say, well, even if I don't want to invent any new chemistry, right, what are the analogs? What are the things that I could try to access? And you can enumerate billions of hypothetical analogs that are commercially available, or the building blocks are commercially available, that would require no reinvention of the chemistry. So this is a little bit concerning, right? It's easy chemistry. We're trying to generate novel structures. We're trying not to change the reactions that we're running. And we can find that we can access many highly similar molecules to a controlled substance, just sort of circumventing the building blocks that are typically used to make it. Sort of one of the realities of molecular design is that it's easy to access large numbers of molecules through relatively simple chemical transformations. Now, we've worked on a number of different approaches to molecular design that sort of use the notion of combining building blocks and reaction transformations. And one of those is sort of shown in this cartoon here. This is a recent model called Synformer. The idea is that we want to teach the model about chemistry. We want to constrain its idea generation using chemistry. And so we will give our model access to commercially available building blocks, right? Things that we can purchase, things that are in stock. And we will give it access to transformation patterns that we have defined and curated. And we will let the model generate hypothetical molecules by generating hypothetical synthetic pathways. So the way that we use this model is we first train it on known molecules with known synthetic pathways, and then we can apply it to sanitize ideas that come from other sources that haven't quite thought about synthesis. So we can take ideas from other generative models that may contain substructures that don't look quite right or that might not be synthetically accessible according to our definition. We can clean them up, right? We can propose analogs that take a structure that we think might be hard to make and convert it into something that we know how to make. And so this sort of generative approach is trying to ensure that any ideas that the models come up with are things that we believe we can take into the laboratory. This is the big sort of thesis behind a lot of the work in our group on generative models, is thinking about making sure that when we get ideas from models, we can actually make them.
Yeah, let's go back to the previous slide. So in one of the slides, you had mentioned that using generative AI, you were getting valid reactions in response to an outcome, but the reaction, you wouldn't go to the red lab at the end of the rolling exam.
Yes, sir.
In this case, how are you sort of overcoming so it's just understanding.
Yes. Yeah. So in this case, we constrain. So basically, you can think about a molecule as the result of a synthetic process. The leaf nodes of this tree, the synthesis tree, we need to constrain those leaf nodes to be commercially available starting materials. That's one constraint. The second constraint is through the templates. We say your chemical transformations must obey these rigid rules. They're approximations of chemical reaction rules. There are no black and white rules in chemistry, really, but we approximate them through the leaf nodes of the tree, and we constrain how leaf nodes are brought together. And that combination gets us to guarantee that anything coming out of the model is something that we know how to make. Yeah. So molecular design sort of offers us a lot of different ways of coming up with structures. So in this case, sort of constrained by synthesis to try to make them actionable. Obviously the use case for this that's beneficial is you make molecules that have useful function. You make therapeutic candidates, you make candidates for material additives. Dual use, obviously, if you think you can invent molecular structures that have property A, you might be concerned that someone will invent molecules that have property B. And so there's sort of these basic concerns of making hazardous chemicals. Also, again, the sort of notion of circumventing controlled substances. You can wonder, right, when it's easy to enumerate billions or trillions of accessible molecules, right, the way that we regulate structures is typically by defining these patterns. And so how do we write that pattern for a DEA controlled substance list that covers billions or trillions of potential new molecules? But there are, of course, again, mitigating factors, so reasons not to be too concerned. This is still really useful technology that should be pre-competitive and exists in the open source, partially because there's a lot of known substances, so maybe we're not so concerned about new ones. And I think the most important thing is this bottom bullet point where, again, when we do molecular design, we are often trying to optimize some target property profile, and this relies on our ability to predict that property profile. And so if we can't predict these properties, there's no reason to think that a generative model will be able to invent a structure that optimizes that property. And so there was this comment article some years ago where they had a property prediction model that was trained to predict the LD50, or the lethal dose of different substances. And they used data from PubChem, so public data on toxicities, basically with LD50. And they predicted, they said they predicted thousands of structures more toxic than sort of VX, nerve gas. which is alarming, right? Thinking about there being thousands of molecules sort of in these chemical databases, relatively accessible, that could have a lower LD50 than VX. Horrible, horrible nerve gas, but it's just a model prediction. And so I take these model predictions with large grains of salt, right? When you do this or predict, bioactivity for drug design, you can run into these situations where, again, your model is confident, but it's not actually accurate. And so there's, again, lots of caveats to the way that we think about molecular design in the context of both beneficial use and dual use. And so the sort of very last sort of set of things that I want to talk about just relates to how we bring these together and how we think about combining them in autonomous laboratories that could, in principle, use these ideas of design, use these ideas of synthesis prediction, and run their own closed loop optimization to try to invent new structures. So in autonomous platforms, the basic idea is that we need some sort of feedback loop where we go from a hypothesis to an experiment to running that experiment, getting some results, and then revising that hypothesis, right? Of autonomy, right? So having platforms that are capable of designing, executing, and interpreting their own results. And you could imagine this in the context of molecules, design a molecule, design a process to make it, test it, and then revise your belief about what its properties should be. When we think about autonomous platforms, I tend to try to divide them into sort of two acts. So I think about both the capability of these platforms and the agency given to them. In this case, we'd refer to if I have some hypothetical platform to do chemistry for me, capability says, what can it do? Right? What experiments are physically compatible with this platform? When I think about agency, I'm thinking about what permissions am I giving this device, right? If it's an automated chemistry lab, what do I let it choose to do? Do I let it choose to do experiments on its own, or do I review everything as a human, right? Supervising this process. And, the intersection of these is basically what are you allowing some experimental platform to run on its own without human intervention? And I think this is the sort of interesting opportunity, but also potential risk, right? When you think about the future of chemistry and the way that AI is going to change the way that we conduct molecular discovery. There's different ways of thinking about automated laboratories and how you can. So I like to draw analogies always to computes. You could imagine, right, there's this trade-off between flexibility and robustness in how we think about computing hardware, right? So CPU is very general purpose, maybe less efficient than if there are particular tasks for which GPUs excel, that you could use those. But GPUs don't do everything CPUs do. And as you go sort of more specific to, really application-specific ASICs, for example, you get really efficient and robust, but you've sacrificed the flexibility in operations. And chemistry labs are kind of the same thing. Right? You define the scope of operations you're trying to do. And if it's narrow, for example, if you restrict your chemical space to peptides, right, we actually are really good at automating that, right? And so you're on luck. You have robust solutions. They're commercial. They've been commercial for a long time. But if you want to automate any type of chemical reaction and you're trying to have as general a purpose as possible, then you're sort of on the other side of this spectrum, and you have a system that's probably not going to be very robust, but it may be very flexible. And so this has influenced how people think about designing these autonomous platforms. And again, not just what they're capable of doing, but then the agency that you give to them, what experiments you let them run on their own. At MIT, sort of towards the tail end, when I was there as a student, we had this demonstration of a robotic flow platform. So this, arm that could assemble flow chemistry pathways. There's a number of different examples from industry and elsewhere of, autonomous labs capable of running their own experiments, right? Some of these are designed for optimization. Some of these are just designed to run, linear experiments that a human will specify. But you have these sort of increasingly complex systems, right? Capable of running these complex workflows and doing their own chemistry, biological testing, analysis, et cetera. And so it's really interesting, in my opinion, to think about how all these developments in AI that we use for designing molecules, designing syntheses, anticipating properties, how the sort of planning side intersects with its execution side. Again, there's really interesting work being done in the sort of hardware automation side, where you have these really complex systems able to do experiments that humans do, but of course run potentially in smaller scale, right, 24/7. And so it's a really big opportunity to accelerate discovery. Now, for autonomous laboratories, this has also generated a lot of discussion. If we do have laboratories that are capable of running their own reactions, should we be nervous about what they're able to run? Now, obviously, there's beneficial uses, accelerating the time to test molecules as therapeutic candidates, et cetera. There's a lot of dual use considerations in some articles about this, and again, discussion in the media, you can imagine there's intentional misuse, but there could also be just accidental misuse, right? It's kind of easy to accidentally mix things that are incompatible that could end up being a safety hazard. You might need some sort of oversight, and especially if you're trying to use tools like LLMs to increase the accessibility of this, perhaps you're going to have operators that don't have the sort of intuition to know what is incompatible and what might lead to these adverse events. Thankfully, I do think that we're at a good point because there's a lot of simple mitigating factors to improve the safety of these platforms. You could simply say humans have to approve experiments. You can limit the chemicals that you give the platform access to. So there's a lot of operational safeguards that help alleviate some of these concerns. But still, growing robustness of automation and the growing robustness of AI planning, these are worthwhile considerations to have. So I'm not going to sort of go through the sort of full summary. I'll flip back to this after acknowledgements. But at the last bullet point here, sort of number 7, I just wanted to mention one consideration that's worth thinking about as we talk about the role of AI for beneficial science, but also as it affects things more broadly. And that is the sort of access models for these tools and for the software. So there's a big functional difference between open access software and open source software as far as who controls it, right? Who can use it, what expertise is required to use it, or cost. And this has been involved in many of our conversations with sort of governmental bodies around even synthesis planning, right? What does it mean to give someone the source code versus have a website that they can use? And how should that influence how we develop this technology further? But want to leave any time for discussion, so I'll just end there by thanking some fantastic group members who have actually done this work, fantastic set of students and postdocs, and for our software output, we also do have a few talented software developers really helping us professionalize and polish our tools. Of course, funding support has also made all of this possible, part federal, but also from generous pharmaceutical and chemical companies. And of course, one important funding agency that I'll sort of make a shout out to for our work in the chemistry space is the NSF Center for Computer Assisted Synthesis, which has really been a fantastic way to bring together folks from chemistry and computer science and really help advance more and more of these tools while having these discussions about responsible AI as well. So thank you again, and thanks for your attention.